The overall goal of this procedure is to monitor changes in mitochondrial calcium concentration in live cells. This is accomplished by first transfecting cells with the calcium indicator protein Ratiometric pericam, which is targeted to the mitochondrial matrix. 1 or 2 days following transfection, the fluorescence microscope and software is set up for live cell imaging. Images of ratiometric pericam expressing cells are then acquired upon addition of a calcium mobilizing agonist. The final step of the procedure is quantitative analysis of the acquired images. Ultimately, results can be obtained that show dynamic changes in mitochondrial calcium concentration through fluorescence microscopy of live cells expressing a ratiometric calcium indicator protein.
The main advantage of this technique over existing methods, such as measuring calcium levels using synthetic calcium indicators, is that ratiometric pericam is genetically encoded, and thus can be targeted to mitochondria or any other organelle of interest.
Hello, I'm Dr. Askar Akimzhanov, a postdoc in Dr. Boehning's laboratory, and I will be demonstrating the procedure today.
To begin the protocol, plate HeLa cells the night before transfection on sterile glass cover slips placed in a standard 6-well culture plate at a density that will be approximately 70% confluent for transfection the following day. The next day, prepare DNA lipofectamine 2000 complexes per the manufacturer's instructions with 4 micrograms of ratiometric pericam mitochondrial expression vector and 10 microliters of lipofectamine 2000 diluted in 0.5 milliliters of Optimem for each well to be transfected. Next, replace the DMEM FBS HeLa culture media in each well.